Hey everyone, welcome to week 4 of Physiological Psychology. This week we are going to focus on synapses and the communication between neurons. Let's go ahead and get started with the concept of synapses. So synapses are specialized gaps between neurons. You may remember from our neuron video that we talked about Ramon Cajal, who was looking at neuron cells, particularly infant neuron cells in slides, and was finding that neurons were actually single cells. Around the same time, another researcher, Charles Sherrington, was making observations in his research with dogs. He was studying reflexes, which are automatic muscular responses to stimuli. If you have ever had this experience where you go into a doctor's appointment and they check your knee by tapping on it with a little hammer to see if your leg responds, that's basically kind of the image I have when I think of reflexes. And we're going to talk more about reflex arcs and Sherrington's work in just a moment. But before we do, let's talk about these two terms, presynaptic neuron and postsynaptic neuron. So a presynaptic neuron is the neuron that is delivering the transmission or the information to the next neuron, which would be the postsynaptic neuron. And again, that's the neuron that's receiving that information. Let's take a look at what we're talking about here. So if you can imagine a neuron, which we talked about in earlier lectures, imagine that this is a neuron, this is kind of extending out to a neuron, and that this is another neuron down here. So this would be the dendrite of the receiving neuron or the postsynaptic neuron, and this would be the axon terminal here of the presynaptic neuron. So this space here is called the synaptic gap. So that's that specialized gap in between the two neurons where communication is happening. So this is the presynaptic membrane of that presynaptic neuron. And here is the postsynaptic membrane of the receiving neuron or the postsynaptic neuron. And here we've got neurotransmitter receptors. And these little dots here are indicating neurotransmitters. We're not going to go into too much information about what neurotransmitters are right now, but that is a topic for this week, which will be covered in one of the next videos. And these little dots here, these little circles here are called synaptic vesicles, but these vesicles are holding neurotransmitters. And when a message is coming through, they release their neurotransmitter, and then that neurotransmitter plugs into these neurotransmitter receptors here in the postsynaptic neuron. So they are chemically communicating with one another. Again, real brief way to explain that, and we're going to go into more detail about neurotransmitters in another video this week. Reflex arcs, coming back to that. So Sherrington, again, was doing some work with dogs, and what he was finding is that when he pinched a dog's paw, that the leg for the paw that he pinched would automatically basically draw back, and the other three legs would kind of extend out to kind of keep the dog steady on those three legs since it couldn't use that fourth leg. So a reflex arc is a circuit from sensory neuron to muscle response. So neurons are separate cells, so a reflex requires communication between the neurons associated with that sensation. Sherrington observed that flexion and extension reflexes are controlled by the spinal cord. The way he knew that is that he took it a step further with these dogs and found that if he severed the connection between the brain and the spinal cord in a dog, that reflex of responding to that pinched paw remained intact. So even without communication from the brain, the dog still had the reflex. So that told him that this process is taking place in the spinal cord. So if you can remember, this is basically kind of like a slice of the spinal cord here, this image. So imagine the skin or the paw of a dog, that pinch happening, that sensation, that sensory signal is going up through this sensory neuron. And he postulated that basically there's communication happening, and that if you remember the term intrinsic, meaning that this neuron here is encapsulated entirely in the spinal cord, that there's this neuron that's sort of relaying that information to the different motor neurons that are carrying that signal to the muscles that need to move. So in this example, for instance, it's showing a leg. If that pinch happened, the sensation's going up here, the intrinsic neuron is saying, hey, okay, this muscle needs to be excited and this muscle needs to be inhibited, for instance. So some of the key properties that he discovered about reflexes are that, one, reflexes are slower than conduction along an axon. Two, that several weak stimuli presented at nearby places or times produce a stronger reflex than one stimulus alone. And three, when one set of muscles is excited, a different set becomes relaxed. So again, think of that dog with 4 legs as that one leg was going up to kind of respond to that pinch. The other 3 were extending out to help keep the dog balanced. So basically, these were based on his observation that there's excitatory information that's being sent to some muscles and then inhibitory to some other muscles. So let's apply this to perhaps basically an experience you may have as a human being. If you've ever pricked yourself with a pin or safety pin or something like that, and pricked your skin, that sensation enters your body through a sensory neuron. And so the idea here is that you have this relay neuron, or again, intrinsic neuron, because it's encapsulated here in the spinal cord entirely. So all of its components, the dendrites, the axons, are all within the spinal cord. That's why it's called intrinsic. So that relay neuron is sending information to motor neurons and telling muscles what to do, basically. So if you touch something hot or you're pricked, more than likely your reflex is going to be to draw back and protect that part of your body. So that's reflex arcs in a nutshell. So let's talk about speed of conduction. So how quick are these reflexes happening? So Sherrington made several observations, and he found that reflex arc times varied, but that the speed of conduction was never more than 50 meters per second. If you remember from our action potential video, research on average showed that action potentials are traveling at an average rate of about 40 meters per second. So reflex arcs are clearly slower than action potentials. So because of this, Sherrington inferred that neurons must be communicating with each other. And he's the one that first used this term synapse. So he postulated that, hey, these neurons are communicating with one another, and there's a slight delay because of that, because they're communicating with one another. So as we talked about just a few moments ago, some muscles are responding in an excitatory way, and some are responding in an inhibitory way. So they're receiving information that they either need to be excited or inhibited. And so these two concepts help us further understand that. We've got an excitatory postsynaptic potential and then an inhibitory postsynaptic potential. An excitatory postsynaptic potential is a graded depolarization resulting from a flow of sodium into the neuron. So if you remember from our neuron videos, neurons are already slightly negatively charged. So when we have depolarization, that means that there's a positive ion, in this case sodium, flowing into the neuron. With the inhibitory postsynaptic potential, We have a temporary hyperpolarization when synaptic input selectively opens gates for potassium to leave the cell or for chloride, which is negatively charged, to enter. If we already know that a neuron is kind of just hanging out at sort of a negative potential, hyperpolarization would mean that it's becoming even more negative. So those positive ions are leaving, the positive potassium ions, or the chloride, which is negatively charged, is entering, making that neuron even more negatively charged. So this little diagram here gives us, kind of goes back to what Sherrington was talking about the speed differences between an action potential along an axon. So again, that's about 40 meters per second. And he postulated that because this neuron needs to communicate with this neuron and so forth, that the speed of conduction through a reflex arc is slower, and that presumably it's being delayed because we've got these gaps in between the neurons where that signal needs to communicate between neurons. So if we go back to Sherrington's different types of studies that he did with the dogs, these two concepts sort of came forward of how these signals can reach the threshold of excitation. If you remember from neurons, our neuron videos, the threshold of excitation is sort of a threshold that has to be met for an action potential to take place. So what he found is that there can be temporal summation, which means several impulses of one neuron over time. So he had found with these dogs that one light pinch that was too light maybe on its own to create that, to reach that threshold of excitation, that one light pinch may not be enough, but if he pinched repeatedly at that same light level, that summation of those pinches could reach the threshold of what's needed to have the reflex respond. So to summarize that, temporal summation is repeated stimuli over time having a cumulative effect. So that pinch, even though it may be light and just one on its own may not reach that threshold, having multiple together would reach the threshold of what was needed to respond or have the reflex. And then the second one is the spatial summation, which is impulses from several neurons at the same time. So coming back to Sherrington's work, he found that, again, coming back to a light pinch, maybe that one light pinch on its own wouldn't reach a threshold that created a response or the reflex response. But having a light pinch here and then a light pinch here and a light pinch here, for example, like in multiple different areas of the paw, that those together, it may be enough to reach the threshold of response. So to summarize that, input from separate locations have a combined effect. So here we've got a graphical way to make sense of what we're talking about here. So again, EPSP is standing for excitatory postsynaptic potential, and IPSP is inhibitory postsynaptic potential, where we would have hyperpolarization, and excitatory again would be the depolarization, which is making it moving more towards positive. So in this example here, we're just going to basically use Sherrington's research again as a way to explain this graph. So let's say he pinched a dog's paw lightly. We might have a slight excitatory response, but it may not be enough to actually create an action potential. Temporal summation of two excitatory postsynaptic potentials looks like this. So in this case, pinching the dog's paw lightly twice. Okay, it goes up a little bit, but still hasn't quite met that threshold that we need to meet, which we know as the threshold of excitation. So if this threshold hasn't been met, there will not be an action potential. So number 3 shows us, okay, well, we're going to pinch the dog's paw lightly 3 times and see what that does. And in this case, you can see it met the threshold of excitation, so an action potential was created. And going all the way up to nearly a positive 30. And then down here, you can see that hyperpolarization, and then it sort of evens back out to a resting potential. And then this is an example of simultaneous excitatory postsynaptic potentials combined spatially. So that this example would be like pinching the dog's paw lightly, but maybe like in 5 different areas. But those 5 different areas together meet the threshold of excitation. So again, we've got an action potential there, the hyperpolarization back to resting potential. And this here would be an example of the inhibitory postsynaptic potential. So we see that hyperpolarization where the neuron is basically even more negative or hyperpolarized, and then it evens back out to resting potential. So I put some little arrows here to show you that this is an example of the spatial summation, and this is an example here of the temporal summation, because the temporal is basically repeatedly one after the other stimulus, and the spatial is showing that these stimuli are happening at the same time, but they're happening in different places and together reach the threshold of excitation.